Hello, I'm Nayeon Kim from Bioinformatics Lab at Yonsei University. Today, I will present my current paper, which was published last month in Gut Microbe. Uh, uh, mouse models are essential for studying how external factors like diet and medications affect gut microbiome. Additionally, mouse are commonly used for FMT, make it important to understand the mouse gut microbiome. Previously, gut microbiome studies relied on bacterial culturing, but replicating the gut environment in vitro is difficult. Therefore, limited number of species can be cultured. Recent advances in computational methods for meagenome-assembled genomes, known as MEG, allow for faster obtaining of microbial genomes. MEG helped discover new microorganisms and expand reference genomes. Then, therefore, I construct the Mouse Reference Gut Microbiome named MRGM by collecting whole metagenome samples, assembling and binning to obtain genome sequences. I also incorporated the additional sequence from MGBC and MIBC databases. After merging and clustering the genomes at the species level, MRGM now include total 1,524 species across 17 phyla. Then we compared MRGM with the previous existing catalog named MGBC and found that MRGM contains 400 more species. For 5 lineage, MGBC underestimated the quality of those genomes and filtered those out because of the limitation of checkcam. However, MRGM includes those genomes, making it a more comprehensive catalog. For the MRGM protein catalog, we predicted protein coding sequence from genome data, which is yielding 4 million proteins at a 19% similarity threshold. The sequences were annotated using GNOME-mapper with many annotated for PFAM and core categories, but only a small portion for geotomes. To improve the database, we use DeepGo Plus, which is CNN-based deep learning approach to add geotam annotation and compare the result with GNOME-mapper. DeepGo Plus annotated a large number of proteins. To validate the reliability, we clustered genomes based on geoprofiles and found that species with similar taxonomy were more clustered into a consistent cluster, which is confirming higher annotation accuracy. We used MRGM to compare the mouse and human gut microbiome and found that only 12% of species in MRGM exist in humans, which is highlighting evolutionary adaptation to different environments. While 70% of mouse gut bacteria are shared at the genus level with humans, only 40% of human gut microbiome are shared with mouse. This may be due to the almost available data comes from lab mouse raised from controlled environment. Then we compare the functional similarity between human and mouse gut microbiome and confirm that most annotated functional terms are shared. We did enrichment test between human and mouse gut microbiome for KKO An enrichment test showed that glutamate transporter genes were more abundant in humans, while bacterial chemotaxis and flagellum assembly genes were more abundant in mice. We found that species in the Colinella genus, which is unique to humans, contain many glutamate transporter genes, which is reflecting higher glutamate consumption in humans. Additionally, genes relate to benzoate degradation, which is commonly used in food preservatives, were more prevalent in humans. This result indicates that humans have more genes to process artificial sweeteners and preservatives, which is highlighting the co-evolution between microbiomes and hosts. For those you who are interested, the database is available for download from the web server. Lastly, I would like to thank my current PI, Insok and Professor Peter Belenky for giving me the opportunity to conduct research abroad during the study of MRGM.